Thank you. Hello, everyone. I'm Yuan, and I'm from Beijing Institute of Genomics, Chinese Academy of Sciences. So today, I would like to talk about a novel computational method that we recently developed for the long-read RNA-seq data analysis, which is named ESPRESSO. Okay, thanks to Xueyi's introduction, so it will be easier for me to talk about this background. So this is a classic textbook illustration showing the process of the DNA over gene to a pre-mRNA and finally to a mature mRNA. So there is one step necessary to produce the mature mRNA. Let me, this laser, yeah, thank you. Yeah, so this step is to removing the introns and joining exons, and we call this step splicing. The process of splicing requires a spliceosome, and many RNA binding proteins are involved in regulating splicing. So there are alternative splicing events in which exons of the same gene are joined in different combinations, resulting in different but related transcript isoforms. In particular, so aberrant splicing only occurs in some disease states, and so some of the alternative splicing events can be detected to diagnose these diseases. And there are several classical computational tools, such as MISO and RMETs, can be used to analyze these local alternative splicing events. And what they do is to map the sequencing read to the reference genome, yeah. And next, they just detecting and quantifying the read, indicating the alternatively spliced introns. So in this example of alternative exon skipping event, the tool will count the read supporting this exon inclusion isoform and also the reads supporting this exon skipping isoform. And these two read counts will be used to calculate this percent splice in psi value to quantify this alternative exon skipping event. There are also transcript level tools, and these tools try reconstructing and quantify the full-length isoform instead of the local alternative splicing events. And one typical example here is cufflinks. Unfortunately, according to a third-party assessment, usually the transcript level analysis don't have a very good performance. For example, so this panel is about their performance on human transcriptome. And as we can see, so the blue dots represent their sensitivity. Actually, none of them achieve a sensitivity higher than 40%. Actually, most of them are lower than 20%. Okay, so all of the tools in the last slide are actually based on the short-read data, such as Illumina. And so as you can see here, so each Illumina read usually can only cover 1 or 2 exons. And we know that so for the higher eukaryotic organisms, such as human, usually each transcript contains 8 exons or more with a total length about more than 1,000 nucleotides on average. So the short-read data are not informative enough to reconstruct the full length. By construct, so if we are using long-read, their read length usually is much longer than short-read and can theoretically cover the full length. However, it should be noted that long-reads also have drawbacks. And so, for example, the higher sequencing area rate in long reads will result in inaccurate splice junctions. As we can see from this table, even the best mapping tool, Minimap2, has an inaccuracy of 6%, which represents the percentage of splice junctions or intron that is not correct. And remember that an average human transcript contains 8 exons and 7 introns. So the percentage of a full-length read that contains at least 1 splice junction wrongly identified could be more than 30%. Okay, so to solve this problem, So we first need to obtain a subset of splice junction that with a high accuracy from the alignment. So we find a very clear and direct way to judge a splice junction is whether the basis around a given splice junction is perfectly aligned. So for example, the left panel shows that if there is no insertion, deletion, or mismatches around a given splice junction, then the inaccuracy of the splice junction is very low, only about 0.2%. By contrast, so if there are some indels or mismatches within the distance, then its inaccuracy could increase by almost 2 orders of magnitude to about 10%. And in the raw alignment output by the mapping tool, we can easily find this perfectly aligned splice junction, which are labeled here as colored connections. And by merging them with the annotated splice junctions, we can obtain a high confidence set. And next, we use this set to correct the other low confidence splice junctions that are not perfectly aligned. For example, so this first read, its second splice junction is imperfectly aligned. So we look for the candidate splice junction from this high confidence set that are close to this one, and actually we can find 2 in total. So which one of the 2 is correct? What we use is a statistical model based on the sequencing error. We assume that so the sequencing error frequency and types are similar on the same read. So we can calculate the probability of each candidate using this multivariate hypergeometric distribution based on the number of mismatch, insertion, deletion, and matches. And next, we can just calculate their probability and choose the one, select the one with the highest probability. Ideally, by doing this, we can identify all of the splice junctions correctly and find all of the transcript isoforms. Okay, there are also incomplete long reads, which are sometimes consistent with more than one transcript isoforms. So we use an expectation maximization step to assign them, which will help us to accurately quantify all of the transcript isoforms. So we just submit the source code of ESPRESSO to GitHub, and everyone can download and use it for research. So it's just like its name. It's like a coffee machine. You can use it to process the coffee beans of long-read RNA-seq data, and you can obtain a high-quality transcript isoform. So after we developed ESPRESSO, we will need to evaluate accuracy at read level. So as we can see here, so after the correction of ESPRESSO, the percentage of dark blue increases, which represent the reads with all of the splice junction correctly identified. By contrast, the percentage of red decreases, which represent the reads with at least one splice junction wrongly identified. Of course, we care more about the transcript level performance. So the x-axis here represents the percentage of randomly selected annotation given to ESPRESSO to compare its performance with the other 2 well-known tools, Rentai2 and Flames. As we can see here, so all of the tools have a better performance with more annotations, but ESPRESSO outperforms the other 2 in terms of both sensitivity and precision. Next is about the accuracy of quantification. So the x-axis here represents the relative concentrations of synthetic RNA transcripts, which is the ground truth. And the y-axis represents the read count output by each tool. So as we can see, among all of the 6 tools, ESPRESSO is the only one showing correlation above 0.9. So this example is about a CD44 gene in 2 cell lines of a prostate cancer, PC3E and GS69. So as we can see, there are several different transcript isoforms in PC3E, and this dark blue one has a relatively high proportion compared to the others. However, in the GS69, there are only a few transcripts, and actually, the dominant isoform is this light purple one. So what's more, there are 8 exons expressed in PC3E, but not in GS69. And using ESPRESSO, we can identify 3 isoforms containing these 8 exons. And the only difference between these 3 isoforms are just a 3 nucleotide difference in 2 alternative splice acceptor sites. So actually, we can use Illumina sequencing, which has a higher accuracy to validate this 3 nucleotide difference, which suggests that ESPRESSO has a nucleotide level accuracy, even for the transcript level analysis. In addition to the method, we also generated a large cDNA nanopro datasets, which includes more than 600 million long reads from 30 different human tissues. And we believe that it could be an important resource for human transcriptome study in future. And this study, most of the work was done when I was a postdoc at the Children's Hospital of Philadelphia. So I would like to thank the PI of the lab, Professor Yi Xing, and also the other co-authors of this work. So I have established my own lab. So there are novel computational methods developed by my PhD students about the circular RNA analysis. And they have their posters today. So if you are interested, please go talk with them. Thank you. That's all.
Thanks, Dr. Gao, for his presentation. Due to a time limit, we can have maybe 1 or 2 questions. Does anyone have questions from our audience? We don't have question on that. Do we have any question from our audience? Okay.
Okay.
There is okay.
Thank you.
I'm quite interested about, because there's known that there must be much higher error rate in the long read sequencing, and how can the ESPRESSO deal with those high error rate to like eliminate all those error rate?
Yeah. So for long read sequencing data analysis in the transcriptomics, the major impact from the high sequencing error rate is about this inaccuracy of splice junctions, because we know that. So the reason we want to reconstruct the full length of the transcript is that there are a lot of splicing events. Right? So if we cannot accurately know where is the splice junction, so it's actually so that the output of our analysis won't make too much sense, because for example, if we want to know the protein sequence from this mRNA, we will need to know every single nucleotide on it, right? So if there are a shift of a nucleotide, it's an open reading frame will change, right? So then the protein sequence will be changed. So that's why we need to accurately know the splice junctions. So because of the because of the high sequencing error rate in long read, so they have a higher inaccuracy of splice junction, and that's why we just use this method to judge the splice junction first, so we can get a high accuracy subset of splice junction, and by using this to correct the other splice junctions, finally we can get all of the splice junction accurately identified and quantified.
Okay, so that means you must have a like quite accurate reference genome, so you can
Yeah,
apply
sure.
to like
So
human, mouse, those mother organisms.
Yeah, both the ESPRESSO and the other tools mentioned by Xueyi, I believe, are based on the reference. Yeah, I know that there are also other researchers are developing method for the reference-free background. Yeah, but this work is not reference-free.
I see. Thank you.